Assalamualaikum everyone. Today I will be discussing the lecture number 15, where we have left chapter number 14, lecture number 14 on this. Let's start. The section of the receptors by mechanisms. Recombinant DNA technique has provided insights into their molecular mechanism. For example, binding of glucocorticoid hormone to its normal receptor protein relieves an inhibitory constraints on the transcription stimulating activity of the protein. For example, insulin depicts the molecular mechanism of glucocorticoid action. In the absence of hormone, the receptor is bound to HSP90, a protein that appears to prevent normal folding of the several structural domain of the receptors. Binding of hormone to the ligand binding domain triggers release of HSP90. This allows the DNA binding and the transcription activating domains of the receptors to feed into their functionally active conformations so that the activated receptor can initiate retranscription of the target genes. The mechanism used by the hormone that act by regulating gene expression has two therapeutically important consequences. All of these hormones produce their effects after a characteristic lag period of 30 minutes to several hours, the time required for the synthesis of the new proteins. This means that the gene active hormones cannot be expected to alter a pathological state within minutes. For example, glucocorticoids will not immediately relieve the symptoms of acute bronchial asthma. The effects of these agents can persist for hours or days after the agonist concentration has been reduced to zero. The persistence of effect is primarily due to the relatively slow turnover of most enzymes and proteins, which can remain active in cells for hours or days after they have been synthesized. Consequently, it means that the beneficial or toxic effects of gene active hormone usually decrease slowly when the administration of the hormone is stopped. Ligand regulated transmembrane enzymes inducing receptors tyrosine kinases. This class of receptor molecules mediates for the first step in signaling by insulin, epidermal growth factor EGF, platelets derived growth factor, and atrial neurotransmitic peptide is transforming growth factor, and many other trophic hormones. The receptors are polypeptide consisting of an extracellular hormone binding domain and a cytoplasmic enzyme domain, which may be a protein tyrosine kinase, a serine kinase, or a glucine cyclase. In all these receptors, the two dominants are connected by a hydrophobic segment of the polypeptide that crosses a lipid bilayer of the plasma membrane. The receptor's tyrosine kinase signaling pathway begins with binding of the ligand, typically a polypeptide hormone or growth factor, to the receptor's extracellular domains. The resulting changes in receptor conformation causes two receptor molecules to bind to one another, which in turn brings together the tyrosine kinase domains, which becomes enzymatically active and phosphorylase one another, an additional downstream signaling protein. Activated receptors catalyze phosphorylation of tyrosine residue on different target signaling proteins, thereby allowing a single type of activated receptors to modulate a number of biochemical processes. Some receptors, tyrosine kinase, form oligopolypeptide complexes larger than the diametric meters upon activation by ligand. pharmacological significance of such higher order complexes is presently unclear. Insulin, for example, was a single class of receptors to trigger increased uptake of glucose and amino acids, and so to regulate metabolism of glycogen and triglycerides in the cells. Similarly, each of the growth factor initiate in its specific target cells a complex program of cellular event ranging from altered membrane transport of ions and metabolites to change in the expression of many genes. Inhibition of receptors, tyrosine kinases are finding increased uh, neoplastic disorders in which the excessive growth factor signaling is often involved. Some of these inhibitors are monoclonal antibodies, for example, citramab, which bind to an extracellular domain of a particular receptor and interfere with binding of growth factors. Other inhibition are membrane parameters, small molecules, chemicals, for example, itokinase, which inhibit the receptor's kinase activity in the cytoplasm. Thank you very much. Inshallah, we will be discussing more topics in pharmacological lecture number 16. Stay tuned and subscribe to my channel. Take care. Allah Hafiz.